I think I should have changed my title to Botnets with the Genome, and I would have had a bigger crowd. I'm Mark Adams. I'm a bioinformaticist. I lead the bioinformatics group at Booz Allen here in DC, where I'm a principal. This is essentially a talk that is going to be orthogonal to a lot of what I've been hearing at the conference. It's an attempt on my part, sort of part of a long-standing attempt, to engage the cryptology community, the hacker community, the people on the outside, and get them thinking about some of the really hard problems that we're facing dealing with the data that's coming from the Human Genome Project and others. I think it's really interesting, and it's been an interesting journey for me, and I think it'll be an equally interesting journey for other folks. I do say, and I always parenthesize this by saying, I am not a cryptologist. I don't play one on TV. You will soon find out I know absolutely nothing useful about cryptology. But nevertheless, watching the conference today and yesterday, reading about cryptology for a long time, it has struck me how useful these approaches can be for helping us start to think about how to evaluate and make use of this data. And there's an interesting twist at the end, because it turns out that biology probably has a place to play in cryptology as well. So what I'm going to talk about today, I'll give a little bit of background, a little bit of inspiration, kind of why this is interesting and worthwhile, and why this is worth your attention and time. I'm going to give the world's fastest biology lesson. It's probably too fast to be truly useful, but at least I'll define some terms so it won't be crazy. DNA is data, right? And in fact, I watched the Android exploit talk just before this and was taken by how similar what you do in trying to break a device looks like what you do trying to break into and figure out what DNA data is and does. I'll talk a little bit about the toolbox. What's cool about that is that almost all of the tools are freely available. In fact, this may be a unique field, or at least close to it, where the best tools, the ones that are the most widely used, are actually all open source, freely available and on the net for the most part, as is almost all of the data, very close to it. I'll work through a very simple example. Again, it's hopelessly primitive compared to some of the things that you'll see at this conference, but it should at least illustrate how these things map onto one another. A brief interlude on DNA computing, working the other way, and then just a little bit about how to get started. If, as I hope, one or two of you guys think this sounds interesting, then you'll learn how to get started. So as I said, a bit of inspiration. Look, the Human Genome Project is the moonshot of our generation. Every generation has big science projects. They're cyclotrons, they're moonshots, they're taxonomies. It's coming up with the fundamentals of mathematics at the turn of the century, turn of the previous century. All of these are out there. Ours is about the collection and analysis of the genes, the DNA data, from a whole range of organisms. And we use the term genome a lot, so I'll just define it. Probably everybody in here knows what it is. But a genome is just the collection of all the genes in a given organism. So you say, oh, well, the genome. Well, the human genome is all the genes that would be present in a person. But you might also talk about a given viral genome like HIV. Much shorter, by the way, but that's the collection of genes there. So it's not a deep term, but you'll hear me use it a lot, so I want to define it up ahead. But we're past the human genome and a lot of mammalian genomes, and we're starting to work our way down into lots of other things. And being able to do so can provide a platform for solving some really important problems, and I'll talk about that in a minute. But as I said, what's cool is that analysis of the genome is a deeply collaborative activity. It is something that's carried out in a remarkably wide range of areas, from pharmaceutical companies through universities, through literally, we know high school kids that are working on this stuff and doing very, very surprisingly advanced stuff. Big science project, I don't think there's a case in which the big science project, like the Genome Project, has been so open to widespread collaboration and really inviting. And not to push the analogy, that kind of collaboration and openness is kind of in the genome of the Genome Project. It was designed that way. It's not an accident. A lot of science, maybe most science, especially when one thinks about the life sciences and pharmaceuticals and so on, is really closed. It's stuff that's conducted in a single company or in a single research lab. The Genome Project has really unfolded and opened that out and opened it to widespread participation. But getting there, right, what is the genome? And we'll talk about this in a bit more detail, but getting the genome and being able to take it apart and being able to sort of figure out not just what's the content, what are the strings, but really figuring out how it works really is an effort to reverse engineering. And this is familiar to anybody who reverse engineers consumer devices, anybody who breaks codes. You're essentially taking something that does something well-defined, but the inner workings are not known, and trying to sort of suss out what those inner workings are. And in fact, there are a lot of sort of analogies, and these aren't just sort of topical analogies. These are actually fairly tight. Between devices, you have networking protocols, means in which a device talks to another device. Well, we have that in biology. Within a given organism, each of your cells communicates with other cells via a well-defined set of networking protocols. And we don't know them all, but they're certainly biologically well-defined. Similarly, intracellular pathways. When you're within a cell or even between sets of cells, there are pathways of metabolism that do the biology, that wet stuff that makes biology work. It's really probably analogous to wiring, device wiring. And you'll even see some pictures of this. Even kind of looks that way. This thing does this, and then it goes here and goes here. It look like circuit diagrams. And then finally, thinking a little bit about design drawings, right? Well, every organism actually has the complete set of specifications for itself in its DNA. That's how we can breed. This is how we reproduce. Now, there are a lot of other things. I mean, we could keep going on this. I ran out of room on the slide for the picture. But these things are subject to the exact kinds of exploits that we use to break codes. Very, very similar, and working with very similar data. This kind of collaboration, right? I mean, this isn't a new thing. The idea that, oh, well, biologists might go outside their discipline to get help, especially in this kind of thing. Biologists are not, and apologies to anybody who is, biologists are very rarely cross-trained in mathematics and computer science. It's happening increasingly now. When I first started in this field, it happened absolutely not at all. I was one of the few people that I knew who had both training in computer science and mathematics and training in biology. It just didn't happen very often. But it didn't happen very often 100 or more than 100 years ago. A guy on the left, you may know of if you ever took high school biology. This is a guy named Reginald Punnett. Reginald Punnett was the guy who invented the Punnett square. Remember the Punnett square he did in biology? His job, basically, at the turn of the century in the early teens, was rediscovering Mendelian biology. Remember, Mendel had done the pea experiments and sort of discovered genetics, and then it was lost until Punnett sort of figured it out again. Problem was, Punnett and his cohort couldn't figure something out. And again, for all of you who've taken high school biology, you remember dominant and recessive genes, right? Dominant genes are the ones that traits show up in the population. Recessives are the ones that don't, or they're masked in an organism that has both. Well, they kept asking themselves, well, why are these recessive traits around? Wouldn't the dominant ones just be dominant and they'd all go away? This was a real puzzle. This was a paradox at the turn of the century. Well, it happened that Punnett here was cricket buddies with this guy, G.H. Hardy. He was probably the foremost mathematician, or one of the foremost mathematicians in Cambridge in the UK at the time. But like I said, they were cricket buddies, and they'd play cricket, and then they'd go drinking afterwards. And at one point, Dr. Punnett told Dr. Hardy the problem that I just described to you. Dr. Hardy said, well, there's an easy way. He was almost embarrassed to say, well, this is a trivial application of the binomial theorem. And he sketched it out on a cocktail napkin. Punnett grabbed this and ran with it. Because from his perspective, what Hardy, who was a pure mathematician, not an applied mathematician, thought was just, look, everybody knows that. He didn't know that. And he ran away with this, which is now called the Hardy-Weinberg theorem. You probably heard it if you've done any biology. But basically, it explained why there's this equilibrium in the population. The mathematician, the biologist working together. The kind of the comical end of this story is G.H. Hardy was famous for being a pure mathematician. In fact, his whole kind of job was pushing away from applied mathematics and saying, look, pure mathematics is better and more important. So it was literally an irritation to him until his dying day that he had a theorem, basically trivially derived from the binomial theorem, named after him. And furthermore, one that was in applied mathematics. So be careful when you get involved with biologists, because they may get very excited about your help. But beyond theory, right? Beyond theory and thinking things through, why do we do this, right? Why is this worth this effort? Emerging diseases is a key topic, right? Everybody's heard of things like flesh-eating bacteria, streptococcus infections, emergent tuberculosis, polio coming back, et cetera. Everybody gets really worried about this, and rightly so. But it turns out there are relatively easy ways to attack these diseases. And the way you do this has been done extensively by HIV. I mean, I was an HIV researcher before I started doing informatics computational biology. HIV was considered an intractable disease. You couldn't attack it. And since it worked on the immune system, AIDS was impossible. You couldn't fight it. Well, it turned out by comparing the genome of the virus, HIV in this case, and your genomes of people, you could find differences. I won't go into the details, but it just turned out that a gene called reverse polymerase is a gene that people don't have. And you could make drugs like AZT that attack it. And by varying those drugs to the mutants that appear in the population, you could make a drug that, in effect, will keep you from dying from HIV. That's a really, really neat trick, but it turns out that trick can be used widely. When you've got the genome of both organisms in question, it's not impossible to suss that out. And that's happening now. In fact, if you go look at the patent database right now, over the last just a short time, right, we've only had the human genome in complete state for a few years, but already the patent database is full of these things. That's one key. Another key, energy, right? I mean, I think everybody around has heard about biofuels, and of course, everybody also knows that current biofuels are woefully inefficient, right? The use of ethanol as a fuel from things like corn stock turns out you might as well have just used the gasoline because it's only about 85% efficient to the power you put into it. But it turns out that you can engineer organisms to do better than that, and even do things that are fairly interesting, like butanol. Is not something that a bacteria or a familiar bacteria or yeast normally make, but it turns out butanol is a pretty good gasoline analog, a little bit more power in it than ethanol, but it's also hydrophobic. It's more like gasoline. It can run in a gasoline pipe without having to be mixed with gasoline, for example. That is something that one could, by scrutiny of the genomes, find the genes that are associated with it, and then work backwards to either selecting new organisms or modifying them to produce it. Same thing is true for algae. Algae makes oil, but not very much. Well, algae's good. Sunlight makes a little CO2, makes oil. Maybe sunlight and a little CO2 can make butanol. People are working that now. Even as a trivial case, this is something I've worked on a bit, engineering light production into algae. Well, maybe you shouldn't bother going through electricity. Just re-engineer some of these algae that naturally glow to do so in a more controlled and more powerful manner, and then you just you have glow-in-the-dark bugs. It sounds sort of trivial, but actually it's all relatively achievable and being done. My day job is working on cancer research, sort of the thing I do during the day. Obviously, this is an approach we take all the time. Why is cancer hard to deal with? Cancer is hard to deal with because unlike the microorganism thing I described to you before, where the bacteria genome and your genome are very, very different, right? It's pretty easy to find ways in which they differ and attack them. A cancer cell, unfortunately, is one that is, by definition, almost exactly like your genome. One or two mutations can cause a normal cancer cell to become a cancer cell. However, it's that very similarity, going back through and sort of reverse sequencing that mutation and comparing it to you, can give some illumination on the ways that you attack it. So I go through these things not as just to enumerate a long list, but just to say, look, this is why it's worthwhile. Food, the same story, right? Being able to engineer food can do all kinds of useful things around changing the yield. But lastly, and this is the point I want you guys to take away, why does this matter, right? Why are we reaching out to you folks, for example, and saying, are there new tools we can use to do this more effectively? This is just 2005, right? This is almost 5 years ago. Look at that curve. This is the curve of number of base pairs of sequence organisms shooting straight up, right? I always say into these sort of, when I was first in this field in sort of the 90s, early 90s, this was pretty slow. If you plotted it out linearly, you would have said, oh, well, we'll have a few. We certainly won't get to the human genome by 05 or 07 or even today. But of course, technology's improved, very much like Moore's Law, and you start to shoot upwards. Well, this is shooting straight upwards. We're now expanding outwards into not only sequencing a human genome, or two, hello, Craig Venter, but we're sequencing lots of people's genomes. And in fact, in a very soon, probably within the next 10 years, almost within the next 5 probably, they're going to be sequencing lots and lots and lots of people. Maybe even you. I don't think more than 10 years from today, everybody in this room will at least have the option of having a DNA sequence run that will inform their medical care. Most people think this is going to happen within the next 5 years and maybe the next 10. The point being that we're moving from sequencing examples of pretty much every living thing we care about, and we've got a lot of those now, especially microbes, to saying we're going to sequence all the individuals. Well, what do we do with all that data? Right? What do we do with it? It can't just sit around and we do them one at a time the way we've been doing it. We've got to think these things through in attacks that are more sensible. So, Here's the promised fastest and most incomplete biology lesson ever. I don't know how many people in this room know this already. Just bear with me. If you don't know this, I'm sorry. This won't enable you to pass your biology exam. But I want to just make sure we're all on the same page for terms. When we talk about DNA, we talk about the biology. Well, you know, you maybe go to a doctor and have some blood pulled, or you can even do this at home. You scrape your cheek. From within the cells that are collected, pull out chromosomes. We've all seen these sort of windy X stripy things. Those are actually just wound up coils of DNA, lots and lots of DNA. You know, it's a long strand of it. Each one of those chromosomes has a subset of your DNA. These genes are bits of this DNA packed into the chromosome. That DNA is not only a complete plan for you, but it's also the plans for the how you operate. So. Compressed, and this is always a matter of some surprise to people, it's compressed. It's about the same size as a DVD, right? Actually less than that if you got a double-sided DVD. It's 3.2 billion base pairs, right? It's A, T, G, or C. So think of it as a long bit string with, you know, other than 2 bits, it's got 4. If it's uncompressed, it's 1 ASCII byte per base. Basically, it's 3.2 gigabytes long. There are about 40,000 genes, and it depends on who you ask. We're still figuring that out. This shows you how far beyond we are. But there are about 40,000 genes in that human genome. Also, if you turn to the person to the left or right of you, they only differ in about 1 every 1,000 of those bases. Now, somewhat interestingly is actually other great apes don't differ that much more. And even mice and other vertebrates really don't differ that much more either. Things get really different once you get to microbes. You know, genome is pretty much the same on most people. And I've had people say to me, if we lump-ziv compress the genome, it would be a lot smaller. So, you know, I don't know why biology didn't do that. That DNA now contains all kinds of coded information at a lot of different layers, right? So what does DNA do? Well, it replicates, right? When the cells reproduce or when you reproduce, DNA can make copies of itself. The reason it does that, and I think anybody who's ever seen a NOVA episode knows that DNA is 2 strands. Well, those strands can unzip and get filled in, and you end up with 2, right? It's a fundamental property of DNA. But also, and equally importantly, are some other processes. And these will get important in a second once we talk about the data. But there's something called transcription. Well, remember I said there were 40,000 genes? Well, remember, every cell in your body has the same genome. How are the clear cells that cover your lens different from the cells in your liver or your kidney, right? They have the same DNA. Well, it turns out what makes them different is which of those genes are switched on. Keep that in your mind, because switching networks are really important, and it's a key point of attack in this. But thinking about switching on and off transcription, you make a copy in something called mRNA, which is kind of like half of DNA. For each one of those genes, that mRNA is translated through a process that's very much like assembly language, and you'll see this in a minute. So if you think of this as duplication, you think of this as selection of a file to run. So if this is your DVD or your CD-ROM with all the data, the programs on it, this is selecting which program you're going to run. This is running that program and getting a product. This is essentially a program for making a protein. And proteins do a really interesting thing. They're a long chain of amino acids, simple chemicals, but they fold and wind up, right? And they wind up in a really interesting process. Anybody who's ever had a model airplane with a rubber band in it, you spin the prop around and the rubber band, first it winds up nicely and neatly, and then at some point it goes yoink, it gets all clumped up, or if it breaks. Well, it turns out proteins fold that way, right? They make this long string of stuff and then they wind up and wind on themselves, and pretty soon you get a kind of a glob. Well, it turns out that globification, that 3-dimensional operation, is actually pretty reproducible. The same protein will always turn into the same thing. This is another point for attack, and I'll talk about that in a minute. What these look like, this is a rendering of a, again, freely available program, which I'll talk about at the end of the talk. But you can take a look at those proteins in 3D, and if this were the program, you could spin it and look at it. But these fully formed and operational structures, proteins are the building blocks that actually do the stuff. What makes that clear cover on your cornea different? Well, the proteins, crystalline, coincidentally, turn out that their property is that they're transparent and they line up. So that cell in your eyeball, the surface of your eyeball, knows to make a whole bunch of that. How does it know? How does it know? What's the internal wiring network that says, turn that on? And I'm an eyeball cell, turn that on. This is something, again, which we'll talk about in a minute. So with that, as I said, woefully incomplete biology lesson, We'll talk a bit about how we get this into computable form. Well, there's the DNA again. DNA goes into a sequencing machine, and anybody who knows this stuff will know I'm showing my age. This was hot stuff when I made these slides. That was an ABI 3700 machine, which is now woefully obsolete. But their machines are all kind of like this. They're robots that essentially know how to do a chemical reaction and take a string of DNA and just read it out as a sequence of bits, or bytes, ASCII bytes, which can then be fed into a server and kept and then examined by people using their computers, right? This is the process. And what's happened, and you can tell by looking at that steep upward turn, is these machines have just gotten better and better and better and faster and faster. The first human genome cost $1.5 billion, did it twice, right? One privately and one governmentally. Like I said, somewhere within the next 5 to 10 years, depending on who you ask, your genome, your personal genome will cost $1,000. So the price is falling like crazy. Now this is sort of a surprise point here, which I'll make later. But the analogy: this is what the data looks like straight out of the machine, right? If I'm probably too small to see, but it's just a series of ASCII characters in long lines with some numbers here to make it clear. Actually, this doesn't look too dissimilar to people who use the Emacs disassembler. Looking at the hex code, it's just a big blob of stuff. What's interesting is DNA, and music is always my analogy. Music's just a long linear string of stuff too, right? Just a linear string of sounds. But for people who are musicians, know how to read music and know how to play it, and anybody who listens to it can say, well, there's pretty complex layering of meaning that goes on in music, right? Just as there's complex layering of meaning in text, or Faulkner, or Shakespeare, or whatever. Teasing it out can be challenging, and I'll hit that again, but I want you to take away, both of these look kind of complicated, but when you listen to it, it turns out it actually is something interesting and means something. So this is explicit. Again, I'm really showing my age. This is 6502 assembly language, which is sort of the last assembly language I was actually ever any good at actually writing. Everybody who's familiar with assembly language knows these are the locations, right? So this is the location of a piece of computer memory. So 2800 hex probably. Here is the actual contents in hex of that, 2 bytes. 2800 to 2802 is byte A2 and 41. So this is the actual computer data, but this is what it means, right? So in this case, the opcode A2, load X, opcode 41, decimal 65 says, well, load this amount into X, right? It's a something. So this computer code is interpreted by the assembler to mean a specific thing, right? And when you run this thing, this program, this is a very simple program, but it just generates a alphabet, A through Z, right? So scrutiny of this, if you know 6502 assembly language and know a bit about where these things are going, you could figure out that that's principal language. Well, all right, this is the gene for alcohol dehydrogenase. Here's the location in the genome, and this is actually literally true, right? This is actually that many bases from the beginning of chromosome 11 where this happens to be located. Remember, 21 chromosomes, right? So sort of biochemists sort of arbitrarily break these things up, and they say, well, this is there in the genome. That's its address. Here's the content, right? Just like the data within a within a computer program, you have ATG. Remember, A's, T's, G's, and C's. And it also means something. That's an opcode that means something. It means start making this protein right here. And in fact, it happens that it's also the amino acid methionine, right? Start doing this. Now it's got to start. And now each of these amino acids are coded in turn by these triplet codes. It's just like assembly language. There's a lookup table someplace that looks up this and creates that. And there's a chemical process pretty well understood, which I won't go into. But it makes a long string. Well, there are other kind of commands, right? So these little commands. This tells it to splice, cut out some junk, and attach this to this. So there's a splice start, there's a splice end, and you keep going, putting it together, cutting and splicing, following the instructions until you get here. There's another opcode, this opcode, and there are a few others that mean this end. OK, done, stop, protein finished. Now that protein, as I said, will fold up, and what do you get? You get alcohol dehydrogenase. I picked alcohol dehydrogenase arbitrarily, but if any of you were out late last night, alcohol dehydrogenase is for detoxifying alcohol in your blood. And in fact, there's a whole, I'm sorry. Actually, the truth is it's the ketone format of the alcohol that actually makes you drunk. So if that weren't there, you wouldn't get drunk at all. Yay, yay, okay. Genetic engineering at work, right? I see possibilities. And people think this is boring. So alcohol dehydrogenase here is commanded to light up when there's alcohol in the bloodstream, right? There's a control network. And I'm going to talk about this a little bit later, but I want you to keep this in your mind, right? The switching on of this actually represents a fairly, at least from the perspective of biologically well-established protocols. Alcohol, need alcohol dehydrogenase. I drink, I get drunk. Then the fall down part is later. What that data looks like, and of course, I have to say, what I made here, I did this. I mean, there's no program, I suppose I could write a program, but I was too lazy. There's no program that lays out DNA like this. I just did it because it looked like this, and I thought it would be easy to understand. When you go to the genome databases, and I'll show you about this at the end of the talk, you can fetch the gene for alcohol dehydrogenase, and it kind of looks like this, right? There's some well, what is it? ADH5. Well, that means alcohol dehydrogenase, a chemical representative. I've always thought it was cool that the EC number for that particular gene is 1.1.1.1. You know, it almost looks like an IP address, these things, but in fact, this is how you look these things up computationally. But here's the sequence. This is actually the amino acid sequence, right? But it's all in there, and it's in sort of nice parsable format. Well, it turns out you can also grab it in XML, right? And this leads in a bit to where I'm going with this, right? There's the same, there's all kinds of metadata associated with it, and sequence. There it is. Remember, A's, T's, G's, and C's. There you go, along with all kinds of markup. And this is a very short format. There's a lot more markup you can get fit on the page. The point being, we're seeing the transition now from all that wet, sciencey stuff. This is looking increasingly as we move along like any other piece of code, any other attempt that we might do to collect, catalog, manage, and reverse engineer and ultimately hack a product. But to do that, you need some tools, right? What do the tools look like for doing that? You know, where do you get this stuff? Well, like I said, DNA sequences, protein structures, biological outcomes. You need those things, right? You need some information, preferably in computable format, so you can go run these hacks on it to try to figure out what's going on. Also, you need tools for managing and analyzing and visualizing the data. Now, you could just write them. That's what I did. I spent most of my career when I was teaching at BU writing code for this because there wasn't any. Well, it's nice, actually. There's code for this, and much better than anything I did available. Everything from databases to statistics software. And something I alluded to at the beginning is, somewhat interestingly, the best of this stuff is open source and freely available. And it's not a coincidence, by the way. It's because the researchers who are working on this are improving it really fast. We're learning a lot very quickly. Remember that upward curve? The way that these guys communicate that with their peers is they just get it out there. If you wait around on licensing and packaging and all that crazy stuff, it's slow. So in these hotbed areas where people are collaborating and working together really quickly, it's perhaps not surprising that you see this code. But we can take advantage of that because you don't need to have a government research lab or something to be able to get at this. Anybody can do this, and in fact, many people do. So where do you get the sequences? I mentioned, you can get that sequence like I showed. Well, probably the best place, at least the best place here in the US, if I were with my UK colleagues, there's an analog in the UK, but they're all the same, is Entree, right? This is at the NCBI, which is the National Center for Biotechnology Information. They're right up here in the DC area. This is actually run by the National Library of Medicine, right? I think it's actually a really interesting thing. What's the Library of Medicine look like? Well, lots of stuff. But one of the things it looks like is access to all this really interesting information, right? This is interesting because you see I've even clicked on here. I can search for nucleotide. Nucleotides are the fancy word for DNA. And I can type something in. And I literally, to get that example, I typed in alcohol dehydrogenase, and I clicked go. And it gave me a list of things, some of which contained the sequences that I wanted, right? You can do the same thing for protein. But this is like search, right? I want to go find something, so I type it in and I go find something specific I'm interested in. But if this is Google, right, then this is kind of like Google Maps, right? This is the same thing. This is run by Jim Kent at UCSC, Santa Cruz. And we've all, nobody's calling it this anymore, so again, I'm probably dating myself, but we always called this the golden path. Jim Kent, a complete hacker in his own right, he would fit right in at Shmoocon. He sat down when the human genome was sort of first being sequenced, but hadn't been assembled yet, right? Think of this, right? Take the whole human genome, 3.2 billion bases, or ASCII bytes worth of information, broken up into 500 or less lengths. Now, they all kind of overlap. They're 500 long, each one. And over and over and over, and some have mistakes, and some are just wrong, and some are somebody's dog. I mean, it's really a mess, right? You laugh, but this is actually true. One of the things we spent a lot of time doing, and this perhaps won't surprise people in this audience, is taking away and figuring out which were the E. coli bits that got in there. E. coli is a common human, it's not a pathogen, but it's a bacteria that's very common in people, and it's also a bacteria that's very common in the lab. So a lot of that sequence turned out, well, it's E. coli sequence, or it's Saccharomyces cerevisiae, brewers and bakers yeast. You had to take them out. But at any rate, Jim Kent took all of these 500 long bits, and he applied normal computational approaches. Jim Kent, not really a biologist, he's more of a hacker like the rest of us. He built tooling that put all of that together quickly so people could evaluate it. And the thing he made to look at it was something that we called the genome browser. Jim said, this is the genome browser. Picture this long string here as the genome seen from way above stretched out. And it's a given chromosome. And you can see this here. Anybody who's taken high school biology or seen a chromosome or a karyotype knows that that's a chromosome, and it's got the characteristic striping pattern, and it's got the little centromere and the telomeres at the end. But this here is a picture, and there's a little red dash there, which you probably can't see very well. That's kind of where you are, that you are here. And then it lays this out. And it says, well, at a very high level, you're not looking at A's, T's, G's, and C's at this level. Here's what's there, and some things you can read out about it. The other really cool thing about this is you can lay down a whole bunch of different organisms. Again, this is freely available. This is open. You can just log on to this and do this. Again, like Google Maps, you go to the top level of this and you start really big and you kind of squinch your way down and get a feel for how these things are put together. And this is computationally tractable, exposes an API and so on, being able to do this. And remember I said one of the cool things people do is they try to lay out other organisms, and these, it's even some microbial organisms, against the human ones and see where they differ. In fact, if you can read this here, it says SNP, that stands for single nucleotide polymorphism. Remember the 1,000 or so, every 1,000 or so bases that you differ from your buddy? This lays all of those out. There's some good reasons for this. It turns out that you can correlate those things with all kinds of things, everything from allergies to diseases to responses to drugs. But again, all that information is laid out there. And this is actually pretty cool. And I should have done a movie here. You can zoom down, and you keep zooming down until you get down to the A's, T's, G's, and C's. And you can zoom way back up and look and see how it looks from all the way out like a chromosome. A very, very useful and very interesting tool. And again, it's open. Just take it and take a look at it. Another thing is the protein data bank. Remember, we're talking about proteins, the 3D structures. Well, it turns out that many, many structures, in fact, many proteins now, a good bit of them, maybe even a majority of them, either have a 3D structure known or they've got one that's predicted. And when I mean 3D structure, these are things that would be familiar. If you hack Quake or Unreal Engine, I've seen people take these in and render them. Right? This is just 3D structure. You can apply a few polygons to it and you have a renderable structure. One of the guys that I know in Second Life made a pretty neat statue out of DNA just by importing the DNA file here and rendered it in Second Life. So, the distance between what you're familiar with and unfamiliar with here is not that distant. One of the interesting things, though, is that one can take these 3D structures, even today, and look at how they fit together, just by eye. And I'll hit some programs that do some of this in a minute. But you can pull up a structure. You can say, where are those variations I was telling you about on that? And do they get in the way of things that naturally happen? If that's the case, they get in the way of things that naturally happen, there are ways to develop drugs and so on for that. And they're not even very complicated ways. Sometimes you just look them up. And significant discoveries have been made that way, and I'll allude to it in a second. Well, all right. I've told you where to get data, everything from 3D structures to sequence data. What do you do with it, right? Well, some of these tools are extensions of conventional programming languages. These are things that you're familiar with every day. People hear a program. I've seen programs today in Python and Java. I'm sure there have been things in Perl as well. Pretty much any programming language, Haskell, Ruby, whatever, stick bio in front of it, and there's almost guaranteed to be a toolkit. And again, what these things are is they're toolkits that do that biology stuff, so you don't have to. Things like, well, if this is a sequence of DNA, is there a gene in there? Or sequence of DNA, what is that gene expressed in a protein? Or even, I want to take this 3D structure and render it in Second Life. I mean, almost all of these kinds of things are embedded in these toolkits. So you have your option doing what we used to do and writing them yourself, but this is a reasonable approach. And there are lots of these. And again, every single one of them is freely available, and we have very, very wide and active development communities. But again, if you didn't want to do that, you've already seen one web-based tool, Jim Kent's tool. You can take many of these tools. This is Gene Pattern, developed at MIT. Again, it's open. Gene Pattern lets you build series of workflows around the things. If you had an idea, I have this cryptographic attack, and I'll talk about it in a minute about what we did. And now I want to stack it into a stack of tools that does the rest of the stuff. For example, I want to examine upstream from DNA, and I want to see how often that occurs. That's cryptology. I'll use a cryptographic technique for that. Then I'll put that aside and tell me everything that you know biologically about that. Well, the second part would be really hard if you didn't leverage this. This is a web-based tool. You could import your file and run a fairly simple GUI on it, and you get an answer. In sense with this audience, one of the problems with these tools, this is a local application. This is written in Java. It runs on your programs called GE Workbench. Lets you do things like examine large arrays of data and visualize them. One of the challenges, though, with this group is, unfortunately, the more graphical and sort of GUI this gets, the more you need to know about the underlying scientific principles and the activities, right? It gets harder, somewhat surprisingly. I'll tell you how to address that in a second, but I always like to make the point that although the GUIs are nice, they're GUIs that are designed sort of for scientists in the lab. They make assumptions about what you know and what you don't know. But nevertheless, and as I mentioned before, tools for visualizing 3D protein structures. And in fact, this little visualization here, which I just pasted into PowerPoint, is just a web render off of this tool here. This tool is absolutely the most interesting tool of the lot. And one of the reasons is you look really cool to your coworkers when you're doing this stuff on your computer, because it seems like, I don't know, maybe you're running a meth lab or something. But what I find interesting and shocking, and this is, of course, another example of the way things are driven, computer gaming has absolutely put the pedal to the floor in developing graphics hardware. There simply wasn't, outside of very high-end government labs, when I first started, the software to do this kind of 3D rendering or the hardware to do it on. It simply didn't exist. We literally had to go down the street to our colleagues at one of the national labs to use their systems to render proteins. In, what is this, in 15 years later, every single kid has an Xbox that could out-render that thing 10,000-fold. You can render these proteins. In fact, I should have shown the screenshot of that. There's an iPhone app for, no joke, for pulling proteins, rendering them, and they're meant for scientists. Well, let me show you here my protein. It seems ridiculous, but again, this is what I'm talking about, getting the power out to a much, much broader range of people that just in a very short time ago was solely pharmaceutical companies and so on. So I want to make sure I hit an example, and again, this is where my hideous ignorance of cryptography comes in. But I'll give you an example, and again, I've sort of somewhat tongue-in-cheek called this a man-in-the-middle attack on DNA control. Remember I said genes are turned on and off by switches, more or less literally, right? Not electrical switches, biological switches on the basis of their conditions, right? Something's going on in the cell, you need to turn on the things that deal with it. Alcohol turns on alcohol dehydrogenase. Here's the world's simplest switch. I think probably every high school student learns this now. It was new to me when I was doing research. This is something called lac operon in E. coli. Well, not to belabor the point, but if there's high glucose, these genes are turned on, right? These are 3 genes, right down the DNA. These genes are turned on when you're low glucose and lactose is available, because these genes take lactose and turn it into glucose, right? That simple, lactose into glucose. But you don't want them around if there's no need for them, right? Well, there's a little switch right here, coded for by DNA, that causes this thing called LAC inhibitor. It's a protein that just sits around in your cell. When that protein there sticks to glucose, and it smacks right in the way of the machinery. It seems really silly, right? Well, there's a protein, and it sticks to a certain sequence of DNA, and it just sticks there as long as there's glucose attached to it. When that glucose comes off, it falls off and goes away, and then you can get constitutive expression. You get lots more of it. It's a simple switch. Now, this is interesting to biologists. It was particularly interesting to biologists about 25, 30 years ago as they were teasing it out. The thing that's important for this crowd is that the upstream sequence, just A's, T's, G's, and C's, form a very specific pattern that code for that switch. And in fact, literally, I've done this. If you take that DNA sequence and splice it into another gene right up front, that gene will then be switched, controlled by the presence or absence of glucose. That simple. It is a known fact. Well, the question is, well, let's see. This is network protocol, okay? This is just like packets, right? There's some routing information in packets up front of the packet. Well, in the same way, there's some switching information up front of genes. But if you don't know it up front, how do you figure it out? And we, in fact, don't know all of the switching and coding. We really don't. This is something we don't know today. We took an approach, but this is, and I again, think that people in this audience will think it's kind of silly and perhaps stupid, and if you guys can think of better things, you should be doing it. One takes a DNA sequence and just does a double hash. Pick a word size, hash along, right? Create that double hash and look for extremely common or extremely uncommon words up front of genes. Are they common? Surprisingly common. Now, it turns out that nature doesn't like switches being flipped on and off willy-nilly. If you've got an organism that happens to have those switches willy-nilly, it quickly is outcompeted by its better controlled cousin, right? So after enough evolution, you have this being, this particular set of sequences being very rare or unusually common, depending on how common the switching is, in front of genes. So just by writing what would be probably extraordinarily simple to you guys, a double hash and hashing through the whole genome, and then looking upstream of those genes and saying, is this unusually common? Because if it is, it might be a switching network. It might be part of a network. You've interdigitated yourself. You stuck yourself in the middle of the biology signaling one way and back. And then you ask yourself, like you would with routing of packets, is there something unusual or uncommon going there? Well, it turns out that identifying those words, quote unquote, that are over or underrepresented in the context of a network. Remember, I told you that these things would look like wiring diagrams. This happens to be a network of genes which together respond to a particular chemotherapy agent. Again, this is, by the way, this network here is just downloaded for free off of Biocarta. It's a website that biologists use. But if you take a look at these and you ask yourself, I found a particularly uncommon set of words that happen to statistically associate with this set of genes. They're what's called coordinately regulated, right? Just like the alcohol dehydrogenase or genes that might be associated with glowing in the dark, they go up or down together, right? And identifying how these networks are related, one can provisionally guess as to what the words are by essentially intercepting the networking protocol and associating the findings. Now, of course, the point is, what's the next step? We've created a hypothesis. You try it in a lab. You go mess it up and see what happens. This is exactly like the guy who was talking about the Android hack this morning and other things that we've heard about today and previously. You just go try to hack and see what happens, see if you can exploit it. And it turns out, I mean, that's drug design, really. This is actually a drug called Gleevec from Novartis, which was literally discovered the way I described, literally. And then they did the test in the laboratory and found out that it had a potent anti-cancer effect in certain cancers. So I'm going to take 5 minutes and do a little interlude before I close. But I want to say this works the other way, right? I know somebody named Len Edelman. I think perhaps many of you others do too. Len Edelman's famous for being the ANRSA, right? But he's also famous among my cohort for having gone backwards. Len Edelman thought that maybe DNA itself could be applied for as a computing substrate. And in fact, Len gave the talk at MIT when I was there by bringing a bottle that was about that big. And he said, look, it's a supercomputer. It has 1 trillion processors. Isn't that neat? Right? And we all thought he was crazy. But then he talked through how that worked. And it turns out that's an approachable, tractable activity. His point is, take really hard problems. Solvable, finite time solvable, but really hard. In this case, the traveling salesman problem. Take those problems and code them in DNA. And the way Len did this was pretty neat. He associated a DNA sequence with each of these cities in the traveling salesman problem, right? So each of these cities gets a DNA sequence that's unique and non-overlapping. And then the lines between them representing the accessible paths, he coded essentially by taking half of one city and half of the other city. So you can see this here between 2 and 3. There's a link there, and then half of this one, half of that one put together. This exploits a property of DNA, right? Remember DNA unzips? That's how it replicates. Well, it turns out that in DNA, the A base sticks to the T base, and the G base sticks to the C base. They just stick together. And once you've got a strip of these things, they'll stick together, and they can only be melted at about boiling point. So they really stick together, right? So it turns out if you take this and make this, this will naturally stick this and this together. So this path from here to here sticks this city's DNA to that city's DNA and leaves a bit overhanging, see? And see how these things, the 2 green ones, both stick together on that? That means this city, this path from this city, this path to this city can be stuck together. And there's a process called DNA ligation. So you basically you mix all of these up, all the city DNAs and all the trip DNAs in a bottle. You have them synthesized. I mean, you can do this. It's sort of relatively cheap. It's about it's $3 or $4 a base. You go to the back of Science magazine. It's like going to the comics and getting X-ray specs. You can order these out of the back to your heart's content. You put them in a bottle, you shake it up, you boil it, and then let it settle to temperature. And what will happen is every possible path, up to actually surprisingly long length, will form, because randomly. There are trillions of these. They're all bouncing around at once, and they kind of settle down into their bits. And you run something called DNA ligation that sort of glues them together permanently. and you electrophoresis them. And electrophoresis is a process that separates things by length. It's a chemical thing. You use like Jell-O and electricity. It's easy. You could do it in your kitchen. And you separate them by length, and you take the shortest one, literally. They're going to be bands. There'll be this band, this band, this band, and this band, representing every path that the DNA computer found through there. Take the shortest one, cut it out, and send it to the same company that you bought the DNA material from for DNA sequencing, and they'll give you the bases. They'll give you the list of bases, which you take apart into the answer. Now, you could solve this little one on your personal computer here, but everybody knows the traveling salesman problem, knows it's NP complete, but it gets hard fast. Having all of these, having many, many, many things working on parallel on it can make problems that are not accessible potentially accessible. I will say that this is, again, for those of you who are computer scientists, right, this is demonstrably a Turing-complete machine. In other words, you can build a complete Turing machine using the same approach that's Turing-complete. And we know what that means, right? It means you need a program that you can write, at least conceptually, you can do on a DNA computer. Now, I say at least conceptually because it turns out there's some really hard, messy, complicated things. It's just, I'm a biologist, so I'm just used to messy and complicated. Integrated circuit designers get very upset, and they say, it's stochastic. It doesn't always work. Well, biology doesn't always work, but it usually works, and that's what makes biology go. But it is an important point. This is just essentially pulled out of a very, very good review on this, which is, if you look at the slides, it has the link, about how this can work. Again, looking out to this crowd here, and I mentioned this to one of the cryptography geeks here at Shmoocon, and I said, well, I'm kind of surprised that nobody doesn't take high-value keys that are really hard to get, think like the DVD encryption key, and just do this, right? I mean, it turns out this isn't that hard. It's tedious and difficult, but might provide accessibility to when a single code key is really important to break it down and do it. Unfortunately, it just turns out there's some application problems. This is kind of hard to do. I think that hard to do, frankly, comes more with the fact that, remember I said at the beginning, it's almost a completely disjoint set. Biologists over here and cryptographers and mathematicians over here, and they just don't talk. It's like Hardy and Punnett. Except they play cricket together, they didn't talk to each other. So again, as I said, sort of the hidden purpose to my coming here today is saying, if I could just interest a few people in what's going on here, I think there's some really interesting opportunities in both directions for fruitful collaboration. So I'm going to wrap this up here in just a couple minutes, but I want to point out, you're not limited just to science, right? In other words, everything I've talked about has been very scientific, very biology-oriented. Well, it turns out DNA data is really interesting in and of its own right. As for example, as an artistic material. I have a colleague who does paintings, auto paintings, based on DNA data, which are very interesting. This is something I did myself. Again, remember, custom Python programs. I coded some Python to pull some sequences from Entree. Remember, I showed you this, mixed with the library somebody else wrote to write MIDI out. I took that DNA data, used the DNA data to create a rhythm from the DNA sequence, melody from the protein sequence, remember that, and harmony from the 3D structure. We talked about all these things. Put them all together, feed them into just a little MIDI synthesizer, recorded in Audacity to make music. And what's really interesting is different proteins sound really different. Really different, surprisingly different. And I think partly because we're mapping to things we're familiar with. This oralization, by the way, isn't just for fun and games, although it was entirely meant to be a Burning Man project. But it's not just fun and games. Often, linear strings of data are really hard to read. Remember that big block of text that was completely impenetrable? But listening to it, it turns out that humans are well adapted to listening to long strings and hearing the differences audibly. So that's oralization approach may actually even have some scientific utility. So I'll wrap the talk up by saying, okay, hopefully I haven't bored everybody to death, but B, I've found people who are interested in this stuff. Here's how to get started. If you want to do this kinds of thing, this is what I'd recommend. One, find a buddy, find a biology expert. There's probably anybody listening to this would go, God, crap, that's a lot of biology, a lot of science in there. Well, find somebody who can kind of tell you what the problems are. His or her problems are going to be the things that ought to drive you and inspire you, and they can answer questions. Take some classes, right? Several universities around here actually offer bioinformatics classes. But to some degree, even better than that, I used to teach bioinformatics at Northeastern and BU. Surprisingly, some of the best biology and bioinformatics classes are online. I'll go through a few of them, but I mean, literally, the world's experts on this can be watched for free on YouTube or iTunes or whatever. Talk to your peers, right? If you guys go do stuff that's interesting, get it out there. There are conventional ways to do this, like all of us scientists, we publish papers, we give talks at meetings and so on. But there are also things like IRC, virtual discussions, forums, just like there are for folks in the computer security or in hacking, talking to people and social networking. And then finally, stay current. There's nothing worse, and this has happened to me more times than I care to admit, in thinking you found out something really great, and then you go read and realize that it was done not that long ago, or worse yet, it was done by Gauss, 200 years ago. So just a sample of kind of these online classes. MIT OpenCourseWare has a series of courses that are really good. I've actually audited this class. But excellent. The video's up. The coursework is there. This is just an example. I mean, it's fit on the slide. There are many more website here. Stanford Bioinformatics Online. These are all from Doug Brutlog's lab. He is one of the absolute leaders in this. And again, his courses with video and notes and everything are also up, and many, many others. Just two that fit. Some virtual places to meet and communicate. I mean, literally, you just go to the Google and type science social networking and see sites you find interesting. I didn't show some of the video sites like Juno and others. Those are cool and interesting and ways to meet and find people. And then, as I said, staying current. More and more of this stuff is actually freely available. It's one of my complaints. It's so much of the work that we do you can't get out. But you can go to PubMed Central at Entree, remember when I showed you that, and find about half the articles these days in my field are publicly available. But there are also freely available journals like PLOS and others you can go to. You can get into a local university library unless you're at Harvard, because they don't let anybody who's not at Harvard into Countway. But pretty much most university libraries will let you in and read material. So that's it. Thanks for sitting with me. Thank you for staying with me. I hope at least somebody is interested to go out there and sort of apply some of what you've been doing to hacking the genome yourself. Yes, please. Will your slides be available? Yes, I'll put the slides up. They'll be on Radio Free Genome. So if you put Radio Free Genome into Google, in fact, I think they're linked there now to a news item. I was talking to somebody else about this earlier. It's one of these things where I think, yeah, it's on the ground there. I think molecular computing is one of these things where it turns out that the devil's in the details.